Of Unit 2 in microbiology. Rather than embedding the videos for this section into this presentation, they are available to you in Angel to view outside of this presentation. Each of the videos is approximately 5 to 10 minutes long, so it makes it pretty long to embed it in here. This way you can watch them separately and go through them again if you need to. There are several different stains used in microbiology, but the Gram stain is the most common. Before staining a smear or sample, a thin film of the organism is placed on the slide and allowed to air dry, forming a smear. It is important to allow it to air dry before heat fixation, or the organisms will not stain properly. With heat fixation, the slide is gently passed through a flame with the smear side up. Generally, 3 quick passes are done, and care should be taken to make sure the slide does not get too hot. As a rule of thumb, it is that you should be able to stick that slide on the back of your hand immediately after passing it through without burning yourself. Chemicals can also be used for fixation, such as methanol or formalin for 1 minute. Heat fixation is generally quicker, cheaper, and avoids the use of toxic chemicals in many cases. An acidic dye will have an anionic chromophore and stain alkaline structures. A basic dye has a cationic chromophore and will stain acidic structures. Basic dyes are more common because most cells are negatively charged. Simple stains will use just a simple single basic dye. Some examples are crystal violet, safranin, and methylene blue. Differential stains use more than one dye so that different cells, chemicals, or structures can be differentiated. There is a link here to a Gram stain video that we produced in one of the quarters in the lab. When using a Gram stain, we begin with a slide with the organism already heat fixed in a smear. With practice, you will get to know how thick to make your smear. In the past, chemicals used in this stain were washed down the drain, but many labs are starting to collect the chemicals to keep them out of our water supply. You may choose to wear gloves when first learning to stain, since the dye will stain your fingers. Any of these stains are at least somewhat toxic, so it is best to either wear gloves or use caution to minimize the amount of stain coming into contact with your skin. It is also likely to be a permanent stain on your clothing. First, the primary stain of crystal violet is applied and allowed to sit on the smear for 1 minute. You will want to cover the entire smear, but remember excess dye is very likely to end up on you, so try not to use more than what it takes to cover the smear. Some of the other videos that you can watch online showing doing Gram stains will show using very large amounts of the crystal violet stain. Nearly every time I see students do this, they end up wearing a significant portion of the stain. The second step is to apply the mordant, which is Gram's iodine solution. I'd pour off excess crystal violet beforehand to keep it less messy. The iodine will sit for 1 minute and makes the dye less soluble. The third step is to decolorize. Here, an ethanol acetone solution is used to rinse away the crystal violet for 10 to 30 seconds, and then quickly rinsed with water. During decolorize, you will want to decolorize till you no longer see crystal violet running off, but be careful not to over-decolorize, since acetone will wash the organisms off relatively easily. Finally, the counter stain of safranin is applied for 1 minute and then rinsed off with water. The slide can be blotted very gently to remove excess water, but still needs to air dry a few minutes. If you are short on time, slides can be saved after heat fixing or after Gram staining until your next session. Remember to label your slides so you know what is on them. A Gram-positive organism will appear purple, as you can see in this picture here, because it has retained the crystal violet. The crystal violet is darker than the safranin, which will also stick to the organisms but not show up over the purple. A Gram-negative organism will appear pink, which you may also see in this slide. The crystal violet does not deposit in the wall and washes away, leaving only the pink safranin. When labeling your slides, you may use a grease pencil or a permanent marker such as a Sharpie. The grease pencils are not the easiest thing to find these days and are not always easy to write with, so my preference is to use a permanent Sharpie. Remember, other glassware is recycled, so please do not use permanent Sharpies on the other glassware. You will label it with your name or initials, date, and the name of the organisms or sample number. Be sure to write on the end away from the smear you plan on holding, since the acetone can remove the Sharpie on some of the slides. Slides are generally disposed of in the trash since the organisms are dead and it is not cost-effective to reuse the slides. Gram stain waste is collected in a separate labeled waste container. The acid-fast stain, or Ziehl-Neelsen procedure, is used for organisms with waxy walls that don't stain with simple stains. This is used for organisms like the Mycobacterium and Nocardia. Here we soften the waxy walls to get them to take up the dye. We begin with a heat-fixed smear and cover the smear with tissue paper to retain the dye. The smear is flooded with carbonyl fusion for several minutes while warming over steam to drive the stain into the waxy walls. Next, the tissue paper is removed. The smear is cooled and decolorized with hydrochloric acid of a pH less than 1 so that only the wax retains the stain. Finally, they are counterstained with methylene blue. And this picture here is an example of an acid-fast stain, as well as a link that you can watch afterwards. The endospore stain is used for organisms like Bacillus and Clostridium. The Schaefer-Fulton method is used to drive malachite green into the endospore. After cooling, it is decolorized with water and then counterstained with safranin. This will leave green endospores and red vegetative bodies. The negative or capsule stain will stain the background and leave colorless cells. The stains eosin and nigrosin are used and will reveal a negative charge capsule as a halo around the cell, which you can see in this example here. The flagellar stain uses para-rosaniline and carbofusion and mordants like tannic acid and potassium alum to coat the flagella, similar to applying mascara, so it is larger and more visible. Electron microscopy stains contain heavy metals like lead, osmium, tungsten, and uranium. Taxonomy is the science of classifying and naming organisms. Prior to 1758, names were descriptive and varied scientist to scientist. The Linnaeus Method, developed by Carolus Linnaeus, is based on common characteristics, and a modified version of this is still used today. The nomenclature of the organism is a binomial system with a specific epithet. There are multiple levels of classification, beginning with the 3 domains and going down through the kingdoms, phylum, class, order, family, genus, and species. All of the taxa have Latin names or Latinized names. The species has a descriptive name consisting of the genus and species or specific epithet for the binomial system. The 3 domains are the eukaryotes, prokaryotes, and archaea. These are all based on the types of ribosomal nucleotides. While attempting to identify microbes, initially we can describe their physical characteristics such as the morphology or shape. Sometimes this is enough for identification, but often they appear too similar and we must use biochemical tests to distinguish them. Serological tests, such as agglutination tests, will test for the presence of an antigen while using an antibody. Phage typing can be used where phages are specific for a particular host and are used to determine if the host is present. Phages are viruses which infect bacteria. Nucleic acid analysis can be used to compare nucleotide sequences. Once the initial morphology is established, a dichotomous key can be helpful by using a series of paired yes or no statements to efficiently identify an organism.